Hello everyone, welcome to my science classes. We were doing class 9, the fundamental unit of life, biology classes we were doing. So in this video we will discuss about cell membrane and cell wall. Okay, so let's begin. So we have already discussed the three main components of a cell, that is, they are having nucleus, they are having cytoplasm, and they are having cell membrane. So now in this topic we'll discuss about cell membrane, and next part we'll study about cell wall. Clear? So moving on to the next slide. So these are some characteristic features of cell membrane. Alright. So cell membrane are flexible. They are not rigid. They are flexible. Understanding? And they are made up of proteins and lipids. Okay. They are protecting the integrity of the interior cell. Means inside the cell, whatever matter is present, the organelles, the cytoplasm, the nucleus, all of them, they are protecting. The cell membrane is protecting the integrity, the inner portion of the cell. Understanding? And they are providing support and maintaining the shape of the cell as well. Okay. Giving support, protecting, giving shape, made up of proteins and lipids, and are flexible, you have to know. Okay? Understanding? So moving on to the next slide. So plasma membrane, or we can say cell membrane, are called selectively permeable membrane. Why it is called selectively permeable membrane? See here, they are called as selectively permeable membrane as they permit only certain amount of molecules. Not all molecules can pass through the membrane. Just remember it. Okay. So they are called selectively permeable membrane as it permits the movement of only certain molecules in and out of the cell. Okay. Not all molecules are free to diffuse. All molecules are not allowed. Only few of them are only allowed. That's why they are called as selectively permeable membrane. Understood? Okay, then moving on to the next slide. So there is a concept called endocytosis. Okay, endocytosis. So what do you mean by endocytosis? It is the flexibility of cell membrane that enables. We know we got in the first characteristics of the cell membrane that cell membrane are flexible. So the flexibility of the cell membrane enables the cell to engulf in food in the form of food vacuole, okay, and other materials from its external environment. Such process are known as endocytosis. So in this diagram you can see endocytosis. So it happens in amoeba. So whenever the amoeba see a particular food or something else, so what they do with the help of cell membrane, with the help of the flexibility of the cell membrane, they used to engulf in food, okay, and other materials they used to take up from the external environment. This process is called your endocytosis. Okay, understanding? Okay, then moving on to the next slide. So there is a question for you. What would happen if the plasma membrane ruptures or breaks down? Can you tell me the answer? Okay, whenever the plasma membrane used to rupture or break down, the inner material, whatever is present inside the cell, it will come out and the cell will automatically die. Okay, and if the cell, this will be number one reason. Okay, if the plasma membrane is not there, it's like it got broken down, then there won't be any selectively permeable property. Okay, any material can get inside and go outside. Okay, so let us see the answer. If the plasma membrane ruptures or break down, then the materials inside the cell will come out. Alright, and the cell will be destroyed completely and it will die ultimately. and the cell will not be able to exchange material from the surrounding by the process of osmosis or diffusion. There won't be any exchange. It will lose its property of exchange. Understanding? Ultimately, the cell will die. Clear? So moving on to the next slide. One more question. How does an amoeba obtain its food? So we have already discussed. About endocytosis. So amoeba obtain its food through the process of endocytosis because of the property of the cell membrane, because of the property of the flexibility of the cell membrane. It used to engulf in food. So let's see the answer. So this is the answer. Through the process of endocytosis, an amoeba obtain its food. We got to know as its cell membrane is flexible. Okay. Then food particles are engulfed, forming a food vacuole, and which assists by the pseudopodia. Pseudopodia is the false feet of the amoeba. Okay. Amoeba secrete digestive enzyme for it and undergoes digestion of the engulfed particles once the food is trapped. So this is how an amoeba obtains its food through the process of endocytosis. Is it clear to you all? So moving on to the next slide. The next question is, why is endocytosis found in animals only? Why we can't see endocytosis in case of plants? Why we can't see? Can you tell me the reason? Why it is only found in animals only? The only reason is that in case of animal cell, there is no cell wall, but in case of plant cell, we have a rigid, tough cell wall. This is the only reason why endocytosis is present in the animal cell, because endocytosis is the process in which you need the flexibility property of the cell membrane. Okay. So let's see the answer. This process can only be found in animals because the cell wall in the plants are very rigid, okay, and very tough, whereas animal cell there is no such cell wall present outside the plasma membrane. Understanding the points? Okay, then moving on to the next slide. Coming to the next concept, that is cell wall, which is only present in case of plant cell wall, okay, which are rigid. One property we already got, okay, that the cell wall is rigid, but the cell membrane is flexible. Okay. So moving on to the next slide. So few characteristic features of the cell wall we'll study now. Where to keep it? I'll keep it here only. Cell are different from animal cell due to the presence of cell wall. So in case of plants, we have cell wall. The next point, it is made up of cellulose and are rigid structure. All right. It provides structural support to the plant. It gives a structural support to a plant. Okay. Due to cell wall, cells of plants, so cell wall are present in plants also, fungus also, bacteria also. So it can withstand any harsh climatic condition. They can withstand greater change in the surrounding condition than in case of animals. So any harsh environmental condition they can survive because they are rigid. They provide structural support to a particular plant, fungi or a bacteria. Okay, so I'll bring it above. So example, cell wall enables the cell to withstand hypotonic solution without bursting. So we have already discussed about hypotonic solution. What is this solution? In case of hypotonic solution, the solvent concentration is higher as compared to the cell. Okay, then what will happen? The water will come inside the cell from the surrounding medium. Okay, and the cell will swell. It has the tendency to burst also. So, but with the help of cell wall, it does not enable the cell to burst out because cell wall is rigid. Understanding clearly? See the points nicely. Okay. So can we move on to the next slide? So moving on to the next slide. So there is one more concept called plasmolysis. Okay, so what do you mean by plasmolysis? It is a process. It is a process in which cell loses water. The cell used to lose water when they are placed in hypertonic solution. So we already studied about hypertonic solution. What was it? In case of hypertonic solution, the surrounding medium has less amount of water content as compared to the particular cell. So what will the cell do? The cell will tend to release water from it and the cell will shrink. Getting my point? So it causes contraction and shrinkage of the plasma membrane away from the cell wall. So there is a shrinkage. The cell wall, since it is rigid, the cell wall will remain as it is. Okay, the plasma membrane will shrink. Getting my point? It is reversible process. Getting? It is reversible process means whenever you place the cell back to a normal hypotonic solution, then the cell will regain its size. Understanding? Understanding what is plasmolysis? The concept? Okay, moving on to the next slide, we'll see a picture of plasmolysis. So you can see here the cell has shrinked. The cell wall is remain intact. The cell wall is as it is, how it was here. Understanding? The cell membrane used to shrink, okay, away from the cell wall. That particular thing is called plasmolysis when you place the cell in a hypertonic solution. This process is reversible process also. Whenever there is a shrinkage of the cell and whenever you put the cell in a hypertonic solution, there will be shrinkage of cell away from the cell wall, and when you again put it in hypotonic solution, then again it will regain its size to this one. All right? Understanding the concept or not? Okay. Hopefully you are understanding. Okay, we'll continue the next topic in the next video. So that's all for today. Thank you for your patient hearing.